Okay. So for today, we will be starting about, or we will be starting to discuss the plant development. So basically, this topic would actually cover three major subtopics, which would be the embryonic development, which will be discussed today, and then we have meristems, and then the flowering development and control of flowering. Okay. So, cells and tissues of the plants. So, the plant kingdom is very large, ranging from the algae, and many of which are unicellular, to the multicellular land plants, which exist in enormous variety of forms. Okay, so basically, when it comes to the historical, diversity and of course, evolution of plants, all of which would actually or all of which started with the unicellular algae. Okay, and then of course there is just a part in the lineage wherein these unicellular organisms did evolve to become multicellular land plants. Okay, so by looking into the similarities and differences between the plant and animal kingdom, a study of plant development can actually shed light on the way that development mechanisms in the two groups of multicellular organisms have evolved independently and under different sets of developmental constraints. Okay, so how did we have this evolution of multicellular land plants? It's because, of course, the presence of different factors that are in the environment. So, for example, the presence of nitrogen, phosphorus in the environment that actually cause the changes in the evolution of these unicellular organisms to become multicellular. Okay? So even if the plant has rigid cell walls, all walls and lack of cell movement within tissues, a plant still has levels of biological organization similar to those of animals. Again, compared with your animal cells do actually have a very, very fluid cell membrane. Okay, so the differences that we have for plant cell and animal cells is the difference of its cell membrane and the cell wall. Okay, so the cell wall is more rigid compared to your cell membrane, which is more fluid in in it by its characteristic. Okay, so with that saying, of course, with a rigid cell wall, it would give you the limitations of expanding when it comes to the development or expansion and reproduction of cells in a plant. Okay, so cell fate in development is largely determined by the similar means as in animals. So it's because of the combination of positional signalings. You also have the intercellular communications, extracellular signals, cell surface interaction, and interconnections by cytoplasmic channels. Okay, so these are, the some similarities that plant cells do have, uh, that is also present when it comes to the animal cells. Okay. So the logic behind the spatial layouts of gene expression that pattern a developing flower is similar to that of the Hox gene action in patterning the body axis in animals, but these genes involved are completely different. Okay? So again, if there are similarities when it comes to the cell fates and signals, there is a complete difference when it comes to the application of or the expression of genes or inclusive genes in plants, and of course the different hormones, okay, that are being used for signals. Okay, so such similarities between animal and plant development are due to the fact that the basic means of regulating gene expression are the same in both, and thus similar general mechanisms for patterning gene expression in a multicellular tissue are bound to arise. Okay? So some similarities in the mechanisms of animal and plant development. First, we have what you call the asymmetric cell division. Okay, so as we what we have um, learned before, so some of the cell divisions are actually asymmetrical depending on what type of species, right? So it's because of the presence of the animal pole and um, the vegetal and the animal poles. Okay, so as in plants, the division also, or the mechanisms are also through asymmetric cell division. And then the response to the appositional signals, or the positional signals. And then you also have the lateral inhibition. And of course, the changes in the gene expression in response to extracellular signals. So for the differences in the mechanisms, in some different ways, plant cells can actually communicate with each other, and they arise from different um, parts, okay, of the plant body. And then from the existence of the rigid cell walls and the lack of large-scale movements. So compared to your animal cell, again, more likely for the plant cell, it's the movement of the cells are very limited because again of the rigid cell wall. Okay, and then the fact that environment has a much greater impact on plant development. So again, um, what we see in the environment are actually affecting also the plant development. Ganon din naman sa animals, 'di ba? Meron laging kinalaman um yung environment when it comes to the development, but then again much greater or mas mataas yung role ng environment when it comes to plants. Okay? So additional differences would be the general difference that most of the development happens not in the embryo but in the growing plant. So, kung sa embryo lahat nandun na hanggang magkaroon na ng organogenesis, pagdating naman sa plants, lahat ng makikita nyong differences ay nandun na mismo sa growing plant. Okay, hindi siya dun sa embryo niya nakikita. Okay? So, plant cells are surrounded by a framework of relatively rigid walls, therefore no cell migration in plants, and major change in the shape of the developing plant cannot be achieved by the movement of the sheets of cells. Okay, so kung sa animal cell, more likely the changes do happen when it comes to the transportation or the travel of the embryonic cells, 'di ba? We have the ectoderm, mesoderm, and the endoderm. Dito naman sa plants, wala silang ganun, wala silang sheets of cells. Okay? Para mag-develop or mag-infold man dun sa embryo. Okay? And then when a plant cell divides, it is not pinched half as animal cell. Instead, a new cell membrane and a cell wall is formed in situ, midway between the two poles of the mitotic spindle. Okay? So again, sa animals, you form by pinching in half, but in animals, nagpo-produce sila ng another type of cell. Okay, which has its distinct characteristic of having its own cell membrane. Okay, and cell wall. So in plants, the place of division seems to be determined before mitosis begins, as a circumferential band of microtubules and actin filaments transiently forms in the cortex at the position of the future cell division. And a complete fertile plant can develop from a single differentiated somatic cell and not just from a first fertilized egg. So this is actually we all call the totipotency or the totipotent type of cell, wherein it is capable of giving rise to any cell type or embryo. Okay, so as like din naman sa di ba we have the germ cells that actually give rise to different types of cells. So sa plant, okay, meron silang mga tinatawag na somatic cells. Okay, na nagdi-differentiate. So the mature plant embryo inside a seed is not simply a smaller version of the organism it will become. So all the adult structures of the plant like shoots, roots, stalks, leaves, and flowers are produced after germination from localized groups of undifferentiated cells known as your meristems. Okay? So, 'di ba nga sabi sa animals, lahat na ng makikita mong development ay nandun sa embryo, and then after maipanganak, dun din nagmamature yung mga organs nila. But for but for plants, okay? Makikita mo na lang lahat ng adult structures after ng germination. Okay? So, all of the tissue types in a plant arise from what you call the meristematic tissue. So the meristematic tissue enables plants to grow their entire lives. So it retains cells that never lose the ability to divide and produce more tissues and organs. So ibig sabihin lang nitong mga meristematic tissue na to, continuous yung kanilang growth and development. Okay? Hindi sila nawawalan ng capability to reproduce yung mga cells nila, and of course, kaya nilang mag-regenerate ng mga organs nila. Okay? Di ba ganun naman sa plants, if kunyari may puputulin kang isang stem neto, minsan nagkakaroon ulit ng another limb, nagkakaroon ulit ng kapalit na bud, and then mag-a-arise ulit dun yung plant. Okay? So, two meristems are established in the embryo. The first meristem is at the tip of the root, and the second one would be at the tip of the shoot. Okay, so apical meristem in a meristematic tissue is present in the shoot tip and the root tip. Okay, again, for the at the tip of the root and at the tip of the shoot, you will see apical meristems. Okay, yun yung mga meristematic tissues na nakikita dun sa root at sa shoot. Okay? So, this figure actually shows you the way where the meristem cells continuously replace themselves and also give rise to the different tissue types in the plant, including epidermal, ground, and vascular tissues. So again, sa kanila, meristematic cells, yung parang mga tinatawag natin na mga germ cells. Okay? So kung sa animal, germ cells, sa kanila, meristematic cells. So they are also, or they can also give rise to different types which are found in the plants, which are the epidermal tissues. Of course, this include the covering of the entire plant. So kapag sinabi namang ground tissue, ito yung mga nakikita sa roots, and then yung mga vascular tissues, yung mga nakikita sa gitna, which are the xylem and the phloem. Ito yung mga bundles of tissues wherein it is responsible for the storage and of course the nutrients of the plants. Okay? So, how is sulfate determined in plant development? So, it is determined by the position in the meristem and cell-to-cell signaling. Okay? So, again, bakit position in the meristem? Again, most likely more of the the development happen in the roots, in the tips of the roots and the shoot. Okay? So, kapag sinabing sulfate, ano nga bang mga cells yung dapat ma-reproduce dun sa mga tips na yon? Okay? So, kumbaga nakikita na natin kung pag sinabing sa root, so ibig sabihin more likely the cells that would be reproduced on the roots are what you call your ground tissues or ground cells. Okay? And then of course, meron pa ring cell-to-cell signaling. So the cell wall would seem to impose a barrier to the passage of the large signaling molecules like proteins or extracellular signaling molecules. Ito yung mga tinatawag natin na small molecules or more likely hormones that actually help when it comes to the development of plants like ethylene, auxins, gibberellins, cytokinins, steroids, and peptides. Okay? So, yan yung minsan ng mga hormones na responsible for their growth. Okay? So, I would like you to read the important roles of these hormones. Okay? Basic function nila. Kung para saan sila. Okay? So, steroids and peptides have transmembrane receptors to the protein kinase type, which are very numerous in plants. Okay? So, meron din silang mga transmembrane receptors. So, pag sinabing mga transmembrane receptors, guys, dun sa mismong membrane nila, meron silang mga receptors for them to be able to receive these types of molecules. Okay? So, ibig sabihin, kapag wala tong mga receptors na to, hindi makakapasok dun sa loob ng membrane ng plant yung mga steroids and peptides without these receptors. Okay? So, if you know the membrane transport system, okay, sa animals, 'di ba meron ding mga facilitated na mga molecular transports papunta sa cell membrane ng animals, meron din sa plants. Okay, with the use of these transmembrane receptors. So, the receptors for other types of plant signals are mainly intracellular. So, ibig sabihin nasa loob na sila ng cell. And plant cells also communicate with each other through fine cytoplasmic channels known as plasmodesmata. So, this link neighboring plant cells through the cell wall, and they may be the channels by which some developmentally important gene regulatory proteins and even mRNAs move directly between cells. Okay? So, kung sa mga animal cell, meron tayong mga tinatawag na microfibrils, intermediate filaments. So, for plants, we have naman the plasmodesmata, which connects the neighboring cells with each other. Okay, so basically, this plasmodesmata is very important when it comes to cell-to-cell signaling. Okay, so the size of the channels varies, with those in shoot cells having the largest diameter. Okay, so for the plant development, the model organism that has been chosen for the genetic and developmental studies is what we call the Arabidopsis thaliana, or which is what we commonly call as your tail crest. So in animals, it is equivalent to the Drosophila. It's because of it is actually diploid and has a relatively small compact genome sequence with around 27,000 protein coding genes. Okay? So, ibig sabihin kasi, most kasi guys ng plants natin ay polyploid. So, ibig sabihin marami silang, kumbaga, combination. Okay? Mas marami silang combination of genes to be able to produce a certain or a unique characteristic type of plant. Okay? So, sa Arabidopsis, diploid lang sila. Okay? Kumbaga, kapag sa animal, i-compare mo, meron lang siyang maternal and paternal. Okay? Kasi kapag ang isang animal ay ang isang plant ay polyploid, ibig sabihin pwedeng combination siya ng characteristics ng tatlong tatay o kaya tatlong nanay. Parang ganun. Kasi because of, the different factors that would actually affect yung mga pollination. Yung mga pollination, pwede yan mag-affect sa germination ng isang plant or sa characteristics ng plant, 'di ba? So, ganon. May maraming factors for you to be able to affect a certain type of or the growth of a certain plant. Kaya nga 'di ba, merong mga plants na kunyari ang flowers nila ang dami-daming kulay. Okay? Hindi lang sila nag-i-stick sa isa. So, ganun yung mga polyploid na mga plants. Okay? So, it is an annual type of plant, flowering in the first year of growth, and develops as a small ground-hugging rosette of leaves, from which a branch flowering stem is produced with a flower head or inflorescence at the end of each branch. Okay? So, ibig sabihin ng annual, yung growth niya ay yearly. Okay? May mga annual. Kasi meron din tayong mga tinatawag na plants naman na nagde-depend naman sa seasons, 'di ba? Meron kunyari kapag dry season, sila lang yung nag-grow, pag wet season sila naman yung nagta-thrive. So etong Arabidopsis talagang annual sila. Okay? Meron din namang mga types of plants na wala masyadong kinalaman yung factor of temperature or walang kinalaman masyado yung factor of the presence of water, pero lagi silang nandyan. Okay? Like yung mga ligaw na mga damo. 'Di ba? Lagi lang silang nandyan. Nawawala lang sila kapag dry season talaga, namamatay sila. Okay? Pero meron din namang mga type ng mga grass na kahit naman sobrang init, talagang nagka-thrive pa rin. Okay? So, meron din namang mga types of plants na 1 year dormant sila, yung next year, tsaka lang sila tutubo. Okay? So, depende. May mga factors talaga na nagdedepende. Malaking factor talaga yung environment sa life cycle ng isang plant. Okay? So, it also develops rapidly with a total cycle in the lab conditions of about 6 to 8 weeks only. And like flowering plants, mutant strains and lines can easily be stored in large quantities to form in the form of seeds. Okay, so again, this is the very ideal type of plant kasi nga short lang yung time for it to grow and for experts or scientists to observe how it grows. Mabilis lang siya. So in a matter of 2 months, meron ka na agad makukuhang results depending on the experiment or the experiment that you are doing compared to other plants na kailangan mo silang hanapin after 1 year pa. Kailangan mo silang hanapin kapag ka maulan. Kailangan mo silang hanapin kapag ka summer. Okay, may mga ganong klaseng halaman. Okay? So, for the life cycle of the Arabidopsis, so this image would actually show you how the life cycle looks like. So, in the flowering plants, egg cells are contained separately in ovules inside the carpels. So, makikita nyo yung, if you do the simulation for the flowering or for the fertilization part of the plant development, malalaman nyo yung, kung paano nangyayari yung mga fertilization. Okay? So, fertilization of an egg cell by the male nucleus from a pollen grain takes place inside the ovary. And then the egg then develops into an embryo contained with the ovule coat, forming a seed. So, Arabidopsis is a Dicotyledon, and the mature embryo has 2 wing-like cotyledons, which are actually the storage organs at the apical end of the main axis, is the hypocotyl, which contains a shoot meristem at one end and a root meristem at the other. Okay? So, ito yung tinatawag natin na cotyledons. Ito yung hypocotyl, and then, of course, the root meristem at the other end. Okay? So, yan, dito natin malalaman kapag ang isang plant ay monocot or dicot. Di ba? Pag monocot or dicot, dito sa mga number of cotyledons na meron sila. Okay? So, following germination, the seedling develops into a plant with the roots, a stem, and of course, the leaves and the flowers. Okay, so this is the gametogenesis part coming from an adult Arabidopsis. You will see here the pollen and then the ovules. Of course, it would undergo fertilization. Wherein the pollen grain, diba? Pwede siyang i-transfer dun sa stigma ng plant, and then from there, fertilization would happen. And then, of course, the embryogenesis part, this one, and then eventually it will form a seed. So once the seed is formed, germination would happen, and then of course this type of seedling would actually grow with the cotyledons, the hypocotyl, and then the root. And then eventually it would reach the growth and maturation phase, wherein it would already produce again another adult flower. Okay? So, please take note of the functions of the stamen, the carpels, the petals, and then the sepals. Okay? Make sure to take note of the different functions of the parts of the flower. Okay? So, for the characteristics of the Arabidopsis flower, it consists of 4 sepals surrounding 4 white petals. And then inside the petals are 6 stamens. This is the male sex organ which produce pollen that contains the male gametes. Then the petals, the sepals, and other floral organs are thought to derive evolutionary from the modified leaves. And then at the center of the flower are the female sex organs, which consist of an ovary of two carpels which contain the ovules, and each ovule contains an egg. Okay, so this is an example of the individual flower of an Arabidopsis. So for the fertilization of an egg, so it occurs when first a pollen grain is deposited on the carpel surface grows a tube that penetrates the carpel and delivers two haploid pollen nuclei to an ovule. Okay? So you will better understand this fertilization process through the simulations. Okay? So next, one nucleus fertilizes the egg cell, and while the other fuses with two other nuclei in the ovule, which forms a triploid cell that will develop into a specialized nutritive tissue, the endosperm, that surrounds the fertilized egg cells and provides the food source for embryonic development. Okay, so may part dyan na magpo-produce sila ng triploid and diploid cells. So, following fertilization, after fertilization, the embryo develops inside the ovule, taking about 2 weeks to form a mature seed, which is shed from the plant, and then the seed will remain dormant until suitable external conditions trigger germination. Okay? So, for the early stages of the germination and the seedling growth, it relies on food supplies stored in the cotyledons or the seed leaves, which are storage organs developed by the embryo. Okay, so in a seedling, more likely the nutrients and the stored molecules for ideal for plant growth are seen in the cotyledons. And then Arabidopsis embryos have two cotyledons and belong to the large group of plants known as your Dicotyledons, or usually we call them the dicots. So the larger group of the flowering plant is actually the Monocotilidons. Okay? So, or what we also call the monocots, which have embryos with one cotyledon. And typically, monocots have narrow leaves and include many important staple crops such as wheat, rice, and maize. So, agriculturally important dicots include naman would be like soy, potato, tomato, sugar beets, and most other vegetables. Okay, so dun sa simulations of growing plants, I think you will be using more of the dicots. So hindi tayo lalayo masyado dun sa idea of growing ng Arabidopsis. Okay? So, at germination, the shoot and the root elongate and emerge from the seed. And then once the shoot emerges above ground, it starts to photosynthesize and forms the first true leaves at the shoot apex. So again, when it comes to photosynthesis, please make sure that you know what are the main ingredients of the photosynthesis process. Okay? I may ask those question or that question in your exam. Basic na yung photosynthesis na yan, paulit-ulit na natin siyang naririnig simula pa nung first year kayo. Okay? So, about 4 days after germination, the seedling is self-supporting plant already and flower buds are already visible on the young plant 3 to 4 weeks after germination and will open within a week. So, under ideal conditions, the complete Arabidopsis life cycle will take about 6 to 8 weeks. Okay? So, very, very short lang yung kanyang kumbaga, growth time. Okay? So, for the embryonic development, so embryogenesis occurs inside the ovule, and the end result is a mature, dormant embryo enclosed in a seed awaiting for germination. So, during the embryogenesis, the shoot-root polarity of the plant body or the apical basal axis is established, and the shoot and the root meristems are formed. Okay? So plant also possess radial symmetry as seen in concentric arrangement of the different tissues in a plant stem, and this radial axis is also set up in the embryo. Pag sinabing radial, di ba pabilog? Kapag ka, di ba, kinross section natin ng isang branch ng plant or even a bark of the tree, it's in a circular or radial symmetry. Okay? So the development of the Arabidopsis embryo involves a rather variant pattern of cell division that enables structures in the seedling to be traced back to groups of cells in the early embryo to provide a fate map. Okay? So plant embryos develop through several distinct stages. Arabidopsis belongs to the angiosperm or the flowering plants, which is one of the major groups of seed-bearing plants other than the gymnosperms or the conifers. So nagde-develop sila differently from each other. Okay? So here, the fertilized plant egg cell undergoes repeated cell divisions, cell growth, and differentiation to form a multicellular embryo. So for the first division of the zygote, it is actually at the right angles to the long axis, dividing into apical cell and the basal cell, and establishing an initial polarity that is carried over into the apical basal polarity of the embryo and into the apical basal axis of the plant. Okay, so from here, coming from the fertilized egg, you will see here that it will eventually have an apical cell, okay, on the apical side, and then a basal cell on the basal side. So this would actually give you already the idea of which part of the fertilized egg would definitely grow into the apical meristem and the basal meristems of your plant. Okay? So the basal cell divides to give rise to the suspensor. So this is actually what you call the suspensor. Ayan, yung nasa gitna niya. Yan yung suspensor na tinatawag, which may be several cells long that attaches the embryo to the maternal tissue and is the source of the nutrient. Okay? So the apical cell divides vertically for a two-cell proembryo. Ito yung proembryo, which will give rise to the rest of the embryo. So the next two divisions produce an 8-cell. This is what you call the octant. Kaya siya tinawag na octant kasi 8 yung cell. And into a globular stage embryo of around 32 cells. So this is the globular type. Okay, again, this is the fertilized egg. It will eventually be a proembryo, developing into an octant and then a globular. Okay, so the embryo elongates and the cotyledons start to develop as wing-like structures at one end, while an embryonic root forms at the other side. This is known as the heart stage. Okay, so ito, magiging triangular pa siya. So humahaba din yung mga suspensor, yung depending nga on the number of the suspensor cells, and then magiging siyang heart shape or heart stage, and then eventually magiging torpedo yung shape niya, and then magiging mature embryo. So ganyan yung life cycle ng embryo ng Arabidopsis. Okay? So, kapag okay na yung mature embryo, syempre, it would definitely give rise to a seed, which has a seed coat and then the cotyledons, okay, inside the seed coat. And eventually, it will turn into flower once it finishes its germination process. Okay? So, ayan yung itsura niya, yung cotyledons. You have the vascular tissues, which includes the xylem and the phloem, of course, the ground tissue, and then the cotyledons. Okay? So, again, yung mga suspensor, the basal cells, it will give rise eventually to the root or the ground meristems, and then yung kung saan nyo nakikita yung mga mas maraming cells or yung Yung more likely mas maraming number of cells, yun yung magiging apical meristem nila. Okay? That will eventually form the cotyledons. So, under a microscope, it does look like this. So, nakikita natin dito yung globular stage. Ayan, pag sabing globular, kasi yung apical end niya ay globular or bilog. Tapos, nakikita dito yung demarcations of the suspensor cells. And then, early heart stage, ayan, nakikita nyo na heart-shaped yung kanyang apical end. And then, this is the late heart stage, and then eventually, this is the mature embryo stage, wherein you will already see here the cotyledons, no, developing cotyledons. So, two groups of the undifferentiated cells are capable of continued division are located at each end of the axis, which are known as the apical meristems. So, the meristem lying between the cotyledons give rise to the shoot, while the other end at the opposite end of the axis towards the end of the embryonic root will give or will drive root growth at germination. So the regions between the embryonic root and the future shoot will become the seedling stem or the hypocotyl. So almost all above the ground adult plant structures are derived from the apical meristems. And then the main exception is a radial growth in the stem, which is most evident in woody plants and which is produced by the cambium, a ring of secondary meristematic tissue in the stem. So after the embryo is mature, the apical meristems remain quiescent or dormant or inactive until germination, and seedling structures can be traced back to groups of cells in the early embryo to provide a fate map. So this is the fate map again of the Arabidopsis embryo. This map would actually show you the movement of the cells given the different colors. Okay, so you have here, for example, the octant, you have the green and the yellow colors. So eventually, when it becomes globular, nagkakaroon na rin siya ng parang purple colored cell, which would eventually give rise to your, or which connects the apical and the root meristems. Okay? So, ganyan siya nagde-divide. Okay? Hanggang maging torpedo siya. So, yung mga green nasa taas, nagiging yellow-green yung gitna, and then yellow, may red sa gitna. So, nag-iiba-iba. Okay? Nagdi-divide ng different types of cell. And then, pertaining to the seedling, so yung mga green parts, more likely, they would be, or they would give rise to the cotyledons. The red colored cells would give rise to the shoot meristem. The yellow ones would include the hypocotyl or would give rise to the hypocotyl. And then the orange ones and the purple ones would be part of the root meristem. Okay. So, for the patterns of cleavage naman for plants, up to the 16 cell stage are highly reproducible, and even at the octant stage, it is possible to make a fate map for the major regions of the seedling along the apical basal axis. Again, napaka-importante ng fate maps because yung mga, 'di ba, kulay na nakikita natin, they already give you the idea of which types of cells would actually will be localized in the apical or the root meristems. Okay? So the upper tier of cells give rise to the cotyledons and the shoot meristem, and then the next tier is the origin of the hypocotyl. At the bottom tier, together with the region of the suspensor where it joins the embryo, will give rise to the root. And then at the heart stage, the fate map is clear. Okay? The radial pattern in the embryo comprises 3 concentric ring tissues or meristems. So, the protoderm or the outer epidermis forms the outer protective covering of a plant. The ground tissue, which includes the cortex and the endodermis, fills the interior part of the plant. And then the procambium is the vascular tissue where you find it is found in the center. It transports water and the nutrients within a plant and provides a support. So, sa procambium, makikita nyo na dun yung xylem and your phloem. Okay? So, the radial axis appears first at the octant stage when adaxial or the central and the abaxial or peripheral regions become established. Okay? So, next we have the periclinal divisions. This is which the plane of division is parallel to the outer surface. So, we are already talking about the arrangement of the cells when it comes to the growth of your plants. So, again, the periclinal, it is the plane of division in parallel outer to the surface and give rise to the different rings of the tissues. And then the anticlinal divisions, this is the plane of which cell division is at right angles to the outer surface. So it also increases the number of cells in each ring of tissue. So to better understand, I have here a figure for you to show how the periclinal and anticlinal divisions look like. So again, the periclinal, it's or the direction is actually parallel, okay, to the outer surface. And then the anticlinal is like this, which is or right angle in the right angle, okay, of growth. Okay? So that's how the arrangement of the cells would actually look like dun sa pinaka parang outer portion of the concentric rings. Okay? So, of course, kung merong growth, usually meron din tayong mga tinatawag na mutations. Okay? So, one of the well-known mutations that we have for Arabidopsis is what we call the fast mutation. So, the fast mutation disrupts the regular pattern of cell division by causing cells to divide in random orientation. It also produces a much fatter and shorter seedling than the normal. But although the seedling is misshapen, it still has roots, shoots, and even eventually flowers in the correct places, and the pattern of tissues along the radial axis is maintained. Okay? So, hindi lang fast mutations ang meron um, sa Arabidopsis. Okay? So, again, with this fast mutation, of course, this is a result of mutations when it comes to the gene expressions. Okay? So in this one, it would result you to a fatter and shorter seedling compared to a normal type of seedling. Okay? So next, for the gradients of signal molecule, auxin established the embryonic apical basal axis. So auxin, also known as the indole 3 acetic acid or IAA, is a small organic molecule or hormone that is important with the chemical signals in a plant development and growth. So, ito, si auxin, para siyang, this is considered as a plant hormone. Okay? Kasama siya ng mga nabanggit ko kanina like ethylene, you have the gibberellins that would affect or that is needed for plant development. So, the auxin causes changes in the gene expressions by promoting the ubiquitination and degradation of the transcriptional repressors known as IAA proteins. Okay? So, dito, in the absence of the auxin, this bind to proteins called the auxin response factors or ARFs. To block the transcription of the so-called auxin responsive genes, this includes the genes involved in specifying the sulfates. So, the auxin-stimulated degradation of IAA proteins frees the auxin response factors, which can then activate or in some cases repress these genes. Okay? So, it appears to be acting as a classical morphogen, forming a concentration gradient and specifying different fates according to a cell's position along the gradient. So this image shows you the auxin signaling pathway wherein in the absence of the auxin, the IAA proteins bind and repress the activity of the transcription factors in the auxin response factor family. So if this is the auxin IAA, so you also have here the transcription factors or the auxin response factor, or the ARF family. Okay, so it actually binds with the TC containing DNA sequence. So there's a particular part of the auxin response factor which is being targeted. Okay, so kailangan dun sa protein sequence na yon merong TC na bases, kasi doon nila ina-attack yung protein na yun to stop the translation process. Okay? So auxin targets the ubiquitin ligase complex. So ito yung tinatawag nating ubiquitin. And it attacks the CSF-TIR1 to the OX-IAA protein, causing it to be ubiquinated, and this modification targets the OX-IAA for degradation and thus leaves the repression of the auxin responsive gene. Ayan. So nagkakaroon ng degradation nito. So with this one, this lifts the repression of the auxin responsive gene, which can then be transcribed. Okay. So the earliest known function of the auxin in Arabidopsis is the very first stage of embryogenesis, where it establishes the apical basal axis. So immediately after the first division of the zygote, auxin is actively transported from the basal cell into the apical cell, where it accumulates. So here you will see the role of auxin in patterning the early embryo. So in the left panel, auxin is produced in the original basal cell, accumulates in two-cell pro-embryo, which is the green one. So ayan siya. So the green one is the auxin response and accumulation. In this part, or in this part of the pro-embryo, dito siya lumalabas or nagagamit or being produced. Okay? Through the transport in the basal to the apical direction mediated by the protein pin7, which is located in the apical membranes of the basal and the suspensor cells. So yung purple arrows, this actually denotes or denotes the part of the basal and the suspensor cells. Okay? Yan siya. So there's always this pin 7 that actually helps this or which mediates, okay, the transport of this auxin going to the apical side of the proembryo. Okay, so another pin protein, which is pin 1. So pin 1 is denoted by the red arrow, transports auxin into the developing embryo. So in the right panel at the globular stage, free auxin starts to be produced at the apical pole, and then the direction of the auxin transport is reversed. So kung sa pro-embryo, yung auxin or yung pagmemediate or yung concentration of the auxin is found in the apical end. Pagdating naman sa mga young globular embryo na or yung dun sa mas developed na type of embryo, nagre-reverse na yung direction. So, coming from the apical end, bumababa naman siya papunta dun sa lower end. Okay? So, you have there, in the basal membranes, you have proteins pin 1 and pin 4. So, that transports auxin from the apical region into the most basal cell of the embryo, known as the hypophysis, where it accumulates. Okay? So, naintindihan ba? Again, the concentration of the auxin is in the apical end kapag nasa pro-embryo siya, and it is mediated by pin 7. But once the embryo grows into a globular embryo, yung auxin na yon would eventually be mediated by the pin 4 and pin 1 types of proteins para i-deliver naman yung auxin na yun pababa dun sa basal region ng inyong embryo. So, pin 1, pin 4, and pin 7, lahat sila tumutulong para ibalik yung or i-deliver yung auxin coming from the apical to the basal end of the embryo. Okay? So, other genes included. So, sa animals, we have the homeobox gene. Kapag ka naman sa plants, we have the specified homeobox genes WOX2 and WOX8. So, they are found in the asymmetrically dividing zygote, which is essential for the formation of the embryonic auxin gradient. And both are expressed in the zygote. So, but after the division, WOX2 expression is restricted to the apical cell and its descendants. On the other hand, the WOX8 is expressed only in the basal cell lineage. Okay? So, the importance of auxin in specifying the basal region of the embryo is illustrated by its effects of mutations on the cellular machinery for auxin transport or response. So, other mutations would include the monopteros protein, or the mutations in this monopteros we call MP as well. It encodes one of the auxin response factors you call the ARF5. So, embryos mutant for the MP lack the hypocotyl and the root meristem, and abnormal cell divisions are observed in the regions of the octant stage embryo that would normally give rise to this structure. So, more likely with the monopteros or the MP mutation, nagkakaroon ng problema dun sa hypocotyl and the root meristems. Okay? Next, we have the topless one. It causes dramatic switching of the sulfate, transforming the whole of the apical region into a second root region. So, topless. Nawawala yung apical side nung plant na yon. But then again, it would give rise to another second root region. Ibig sabihin, sa apical meristem dapat apical parts yung mag-grow. Ang nangyayari, nagkakaroon ulit ng isa pang root system. Okay? So, it abolishes the development of the cotyledons and the shoot meristem. And this is temperature sensitive and can be made to act at different times during the development by simply changing the temperature at which the embryos are grown. So, normal function of the topless protein is to repress expression of the root promoting genes in the top half of the early embryo by acting as a transcriptional co-repressor, forming a complex together with OX-IAA proteins and the auxin response factors they regulate. Okay? So, si topless protein daw, siya yung responsible to promote the expression of the top half of the early embryo. But since if there would be mutations on this topless protein, yun nga, nagkakaroon ng repression of the growth of the cotyledons and the shoot meristem. Okay? So next is the shoot meristemless, or the STM. This is a mutation on the gene completely blocking the formation of the shoot apical meristem but have no effect on the root meristem or other parts of the embryo. So, STM encodes a transcription factor that also required to maintain cells in the pluripotent stage in the adult shoot meristem. Okay? So, dito naman, this is another mutation in the shoot meristem. Okay? So, plant somatic cells can give rise to embryos and seedlings. Plants do have an amazing power of regeneration compared to animals, and a new complete plant can develop from a small piece of stem or root or even from the cut edge of a leaf. Yun nga yung kapag ka, 'di ba, gusto nating mag-propagate ng plants, usually ang ginagawa natin, if we have a mother plant, kumukuha lang tayo ng kumukuha tayo ng isang leaf o kaya naman isang stem, tapos itinutusok lang natin sa ground, and then from there, another plant would actually grow. Mag-grow siya ng sarili ng roots. Ganon din nga kapag gumagamit tayo, for example, ng mga cuttings ng vegetables, di ba? Kung na-try nyo sa bahay nyo na mag-experiment netong pandemic na yung edge ng carrots, i-dip nyo lang sa water and then eventually nagkakaroon sila ng roots. So, it's because of the plant somatic cells. Okay? They can actually give rise again to another embryo and of course, a new seedling. Okay? So, propagation of plants in tissue culture. So, usually, ito na yung technique that we are using for us to do propagation of plants. So, tissue culture is the growth of tissue in an artificial liquid or solid culture medium. So, it enables botanists to breed large numbers of plants from somatic tissues, and it enables selection of tissues with desired characteristics. And again, these plant cells are actually totipotent in characteristics. Again, it can become an entire plant because of the quality of the cell. Okay? So, minsan din meron kunyari, luya, kuha ka lang ng isang part ng luya, tusok mo sa soil. After a few days of watering, magkakaroon na siya ng sarili ng shoot. So, ganun yung mga tinatawag natin ng mga tissue culture. We tend to we tend to sever parts of a plant itself or the pinakabunga itself para lang makapag-propagate tayo ng another plant. Okay? So, Meristem culture is the formation of many shoots from a single shoot tip. So, it is for clonal plants that develop when shoots are removed to develop new and genetically identical plants. And then, we also have the somatic embryos that are formed in many steps from mature plants. So, protoplasts are formed by digesting mesophyll from leaves. The protoplast is a cell from which the cell wall has been removed by enzymatic treatment. And then, we also have the callus that forms into clump of cells as protoplast regenerates new cell walls and it divides. And then, callus is manipulated to produce somatic embryos. Okay? So, how do we do this different cultures? So, for example, this is a setup for culturing somatic cells from a mature plant that can form an embryo and regenerate to a new plant. So, from here, you will just get a tissue sample and then place it in a solid agar medium containing the appropriate nutrients and growth hormones. And then the cells will start to divide to form a disorganized mass of cells known as a callus. Okay, so the callus cells are then separated and grown in liquid culture, again containing appropriate growth hormones. So in the suspension culture, some of the callus cells divide to form small cell clusters. And then these cell clusters can resemble to globular stage of a dicotyledon embryo. And with further culture on solid medium, they develop through the heart shape and latter stages to regenerate a complete new plant. So, ayan. Okay? So, pwede kang mag-regenerate just using this one. So, again, in an agar medium, it would be really parang it's a must that you should put appropriate nutrients and growth hormones for this callus to form and to develop into a new plant. Okay? So, next, we also have what you call the Anther Culture. It involves the mature anthers that are cultured in a medium containing vitamins and growth regulators. So, pro-embryos are produced by the division of the haploid tube cells. So, then the pollen grains rupture, releasing the haploid embryos, and haploid plant may be generated. Alternatively, chemical agents can be added to encourage the chromosome doubling, and the resulting plants are diploid and homologous chromosomes carry the same genes compared to the coming from the mother plant. Okay? So, with this one, we have what you call now the genetic engineering of plants. So, traditionally, hybridization or the crossing over of different varieties of plants or species is used to produce offspring with desirable traits. So hybridization is followed by the vegetative propagation of the mature plants to form many identical plants. So it could actually, or this hybridization could actually occur in in laboratory setup, pero pwede din siya na natural hybridization, 'di ba? With the help of pollinators, posibleng mangyari 'yon. Kaya nga nagkakaroon tayo ng mga polyploid na plants. Okay? So today, it is possible to directly alter the genes of organisms, producing new varieties of plants with desirable traits. And we commonly know this as GMOs, okay, or the genetically modified organisms. So with the GMOs kasi guys, this has as far as I know, with the GMOs, they were really used to produce plants for consumption. Kasi nga 'di ba nagkaroon tayo before ng problems when it comes to food production. Nagkaroon ng problema sa food production. So, with the help of scientists, they were able to produce genetically modified organisms wherein nakatulong siya para mapunan yung problem when it comes to the lack of food. Okay, lalo na sa ibang bansa. But then again, these GMO products do have corresponding issues or technicalities. Okay? Kung dapat ba or is it parang ethical to form, GMOs? No, this is a main issue when it comes to genetic engineering. Even naman in plants, even naman in animals, 'di ba? Like the cloning na tinatawag natin, is it really morally and ethically accepted when it comes to the solving issues, global issues, kung ethical ba sila na gamitin just to be able to resolve this issues that we have when it comes to food security, for example. Okay? So, kaya minsan may mga nakikita na kayo na iba't ibang kulay ng corn. Minsan din may iba't ibang kulay ng or iba't ibang shapes of watermelon. More likely, those are already genetically modified organisms. Okay? But then again, we also don't have the idea if, for example, if the nutritive facts or the nutritive components of that certain GMO is still the same with what is, harvested in a natural way. Okay? So, maraming issues when it comes to genetic engineering. But then again, this is actually one of the most successful, for me, one of the most successful things that, that science brought to solve global issues. Okay? So there are actually two methods used for genetically modified plants. So the first one would be the high voltage electrical pulse method and chemical treatments of the protoplast. So there are temporary pores formed in the cell membrane, and these pores enable foreign genes to enter. And then the second one would be the gene gun method, so wherein the cell has intact walls. So the gene gun bombards a callus with DNA-coated microscopic metal particles, and the DNA is delivered into the cell and the nucleus. So these are the two methods they use for GMOs. Okay, specifically for plants. So plants such as corn and wheat have been genetically engineered by these methods. And the plants with a foreign gene is actually what we already call as a transgenic organism or a genetically modified organism. So anything or even if animals or plants yan, basta yung genes nila are manipulated, okay, you call them already as transgenic organisms. So they carry a foreign gene and have a new and different traits. So next, we have the agrobacterium, which can be used to genetically modify plants. This has a foreign DNA, is first inserted into the plasmid of the agrobacterium, which normally infects the plants. And when a bacterium with a recombinant plasmid infects a plant, the plasmid enters the cells of the plant and incorporates into the plant's DNA. So this is an example of how the agrobacterium could actually affect your the portion of the plasmid, which is the tDNA of a certain plant. So again, leaf discs are removed and placed in a culture of medium, of course, with the help of also of the um, growth factors. But then again, there is um, an additional introduction of the agrobacterium containing the recombinant plasmid. So the recombinant plasmid is what you call the tDNA. So genes experimentally inserted to the tDNA, this one, will therefore also be transferred into the plant cell chromosomes. So the TI plasmids modify so that they do not cause tumors but will still retain the ability to transfer the tDNA and are widely used as vectors for gene transfer in the dicotyledon plants. So the genetically modified plants in the callus can then be grown into a complete new transgenic plant that carries the introduced gene in all its cells and can transmit it to the next generation. Okay, so ito ginagamit naman nila si agrobacterium to introduce a different chromosome, okay, dun sa callus na nafo-form. And then eventually, you will be having transgenic plant types that will grow eventually on the medium. So next is the cell enlargement. This is actually a major process in the plant growth and morphogenesis. So, di ba sa animals kasi more likely reproduction lang, but for plants, there are cell enlargement that happens. Okay? So, plants grow in size by a combination of cell division and cell expansion. So, the cell expansion can provide up to 50-fold increase in the volume of a growing plant tissue, and majority is due to the cell enlargement. So the driving force for expansion is the hydrostatic pressure. You call the turgor pressure, which is exerted on the cell wall as the protoplast swells as a result of the entry of water into the cell vacuoles by osmosis. So this image shows you the enlargement of a plant cell. So plant cells expand as water enters the cell vacuoles and thus causes an increase in intracellular hydrostatic pressure, and the cell elongates in a direction perpendicular to the orientation of the cellulose fibrils in its cell walls. Okay, so once water goes in, eventually the plant cells would grow perpendicularly. Okay. So plant cell expansion involves synthesis and deposition of the new cell wall material and is an example of a process what we call the directed dilation. So this is where an increase in the internal hydrostatic pressure causes a distinct change in the shape in a particular direction. So in plants, the direction of the cell growth is determined by the orientation of the cellulose fibrils in the cell wall, and enlargement occurs primarily in a direction at right angles to the fibrils where the wall is weakest. Okay? So the orientation of the cellulose fibrils in the cell wall is determined by the microtubules of the cell's cytoskeleton, which are responsible for positioning the enzyme assemblies that synthesize cellulose at the cell wall. For the leaf development, so the development of the leaf in a particular shape involves a complex pattern of cell division and cell elongation along the proximodistal and mediolateral axis of the outgrowing leaf, which is thought to be organized by a region at the base of the leaf. So cell elongation plays a central part in the expansion of the leaf blade and the latter stages of the leaf outgrowth. So, two mutations that affect the leaf shape by affecting the direction of the cell elongation have been identified. So the first one is the Arabidopsis mutant ungustifolia, which are similar in length to the wild type but are much narrower. So pag sinabi natin wild type, guys, yun yung natural or normal form niya. Okay? But with angustifolia, same yung length nila with the wild type but much narrower. Okay? For the second one, we have the Arabidopsis mutant rotundifolia. The length is reduced compared to the relative leaf length, and of course its width. So neither of these mutations affect the number of cells in the leaf. Okay? So this is the figure showing the differences of the shape of the leaves of Arabidopsis affected by the mutations, okay, of the cell elongation. So again, the angustifolia mutation compared to the wild type, medyo narrower sila, narrower yung leaves nila. And then for the rotundifolia, shorter yung kanilang leaves at fatter. Okay? So those are the differences. So the normal ones is this one, the wild type. Okay? So for the summary of the early development of flowering plants, again, first, it is very important that there is a asymmetric cell division and auxin signaling in the embryo that establishes the apical basal axis. And then next would be the embryonic cell fate is determined by the position, again, with the meristems, apical and the basal meristems. And then of course the shoot and the root meristems of the seedling give rise to the adult plant structures. So the early embryonic development in many plants is characterized by asymmetric cell division of the zygote, which specifies apical and basal regions. Embryonic development in flowering plants establishes the shoot and root meristems from which the adult plant develops, and the auxin gradients are involved in specifying the apical basal axis of the Arabidopsis embryo. One major difference between plants and animals is that in culture, a single somatic plant cell can develop through an embryo-like stage and regenerate a completely new plant, indicating some differentiated plant cells retaining totipotency. Okay? So